All right, welcome everyone. Thanks for joining us today for the Aquatic Invasive Species webinar series. It's 1 o'clock, so we will go ahead and get kicked off now. I have a couple of quick housekeeping items, then I'm going to turn it over to our presenters. We're welcoming today a group from Montana State University. We have Ashley Wolfe and Dr. Greg Chorak on the line right now, and we may be joined also by Ryan Thum later for Q&A pending his conference schedule. They're going to chat with us about management of hybrid watermilfoil. We have about half the time set aside for presentation, and then the rest will be Q&A. We're using a tool called Slido to manage our Q&A. You can go to slido.com, and I'll put the link into chat for you as well. You're going to use the event code MilfoilMapper. And you can enter your questions there at any point. We'll hold them until Q&A, but you can also browse what other people are asking and use an upvote feature to help get the questions that most people are interested in up to the top and most likely to be answered in the limited time that we have. We are recording today's webinar, so you can watch our YouTube channel for that recording to go live. It usually takes about a week or so for us to get it transcribed and posted, but it will be available there, and we'll send out a message to everyone who registered once it's live in case there's anything you want to revisit or you want to share with someone who couldn't make it. And we do have everyone muted for the duration of the webinar. So if you do have questions, we ask you to use the Slido feature. You can also use chat here in Zoom if you're having any technical issues. Michelle Isaacson is on the line as well. She's underneath the AIS Detectors program handle, and she is monitoring and ready to help you as best she can. So I think with that, we will go ahead and get the webinar started. Ashley and Greg, I'll turn it over to you. Thank you.
So yeah, without further ado, we'll get to talking about integrating genetics and herbicide studies to improve watermilfoil management outcomes. So the premise of the research program is genetic variation can influence management outcomes. So individuals of a specific species may respond to different treatments differently. Not everyone's going to respond the same way. Our primary goal is to use genetic information to predict herbicide response and thus improve management outcomes. So an analogy we kind of like to use is the 23andMe analogy, where you can do genetic health screens, or you can ask your doctor for some sort of health screen to look at potential health risk factors that you may be more likely to have, or disease risks that you might be more at risk for. Similar technologies are used, maybe not quite as advanced, but they're used in common terrestrial agricultural weed management. This isn't quite implemented in aquatic plant management yet, but is a goal, and we would like to get there. In agricultural weed management, it's pretty common to have both nucleic acid-based tests and protein-based tests to rapidly test plants in your field to see if they are potentially resistant or susceptible to a particular herbicide you may be using to treat the field with. So this research project has both long-term and short-term objectives and approaches. And I just mentioned this to point out that, some of our short-term objectives and approaches may be overruled or overridden by the long-term ones, but we stick with where we're at and in the short term, I guess. So kind of getting into our study system, Eurasian watermilfoil, Myriophyllum spicatum, is a fully submerged invasive aquatic plant. It reproduces both asexually and sexually. And with that sexual reproduction and somatic mutation, it can create distinct clones, what I'll be referring to as strains throughout the rest of the talk. And those strains can propagate indefinitely through vegetative reproduction across the landscape. It's important to note that being an invasive, it's not native to North America, but Eurasian watermilfoil can hybridize with the native northern watermilfoil found in North America, Myriophyllum sibiricum. And there's a lot of genetic variation because of this hybridization. So what we're looking at here on the right-hand side of the screen is a principal components analysis based on about 2,000 SNPs from across the watermilfoil genome, and where each point on the graph represents a strain or a unique DNA fingerprint. And then the points that are more close together are more genetically similar, and points that are farther apart from each other are more genetically distinct from one another. So we can see these clustering patterns based on the taxon. So we have all of our northerns clumped in one area at the top of the graph in the green. And then we have two different Eurasian watermilfoil clumps marked as Eurasian 1 and Eurasian 2 in yellow and red. And then with these two different clusters, we can kind of infer that these are two distinct genetic lineages. Maybe that arose from two different introductions or two different sources of introduction. And then because we have those two distinct Eurasian lineages, hybridizing back up to the northern, we have two different hybrid, I guess, clusters, you could say. So within all of these clusters, there's a lot of genetic variation, and just across all of the watermilfoil, there's a lot of genetic variation. But the variation that we're most concerned about is genetic variation for herbicide response. So on this slide, I'm going to be talking about the variation for response to fluridone and 2,4-D, which are two commonly used aquatic herbicides to treat and manage watermilfoil. So fluridone is a systemic herbicide that targets the PDS, phytoene disaturase region. And on this figure, each individual panel is a unique watermilfoil strain. And we see on our x-axis is the overall dry biomass. And then on our x-axis, it is either control or treated. And then those are represented by the little dots on the graph. So the left-hand dot is our control untreated, and then the right-hand dot is the treated result of that final biomass. So the box highlighted in red shows a fluridone-resistant strain that, I mean, really kind of likes being treated with fluridone, and its biomass at the end of treatment increases compared to its starting biomass as a control. And then in the green highlighted box, we have a strain that we would consider as sensitive to fluridone treatment, and that is what we would expect to normally see in the field. Something that, you know, is controlled, responds well, most likely dies post-treatment. And then if we look over to the next panel, or the right-hand side of the screen, we're showing an assay done with 2,4-D, which is a synthetic oxinic herbicide commonly used. And on this graph, we're showing two different strains of Eurasian and hybrid watermilfoil, where 2,4-D concentrations on the x-axis, and again, our final dry biomass as on the y-axis. And as we can see, our solid line is Eurasian, and that one is responding pretty well to the 2,4-D treatment, kind of our susceptible response that we would expect. But then the hybrid watermilfoil strain does not respond quite as well, and it looks to be kind of resistant, or would be considered as resistant, especially at those lower concentrations. So throughout the talk, we're going to be talking about these three different levels. The talk will be structured by the taxa and then strains, and then we'll talk about genes, and that's where we'll transition to Greg talking. So with the taxa, we're going to be talking about distinguishing different species. So that's distinguishing Eurasian watermilfoil from northern watermilfoil, and then also their hybrids. And then we go down even further, and we look at the strain level, and then here we're looking at genetic variation for herbicide response within the species, taking into account that many of these plants are highly clonal. We want to pay attention to these strains across the landscape. Arguably, this is kind of our operational unit where we like to focus in at, and where we are currently focused in at in Eurasian watermilfoil management and research in the lab. And then Greg's going to talk to us about identifying specific genes for herbicide response, because the same genes can be present in different strains. So it's important to look even a little bit deeper than strains. So I'll give you the punchline about hybrid versus Eurasian watermilfoil, and then I'm going to walk through some examples to kind of dig that in a little bit deeper for you. So hybrids, on average, grow more vigorously and are less sensitive to 2,4-D treatment. And this is especially true at lower concentrations of 2,4-D. So on this slide, we have two different studies done by past graduate students in the lab. And on the left hand of the screen is a study done by Liz LaRue, where she took a bunch of different hybrid and Eurasian watermilfoil plants from around Michigan and included them in a 2,4-D assay. And then this resulting graph is their dose response curve. So 2,4-D treatment or concentration on the x-axis, and then that overall growth on the y-axis. And as we can see here, our hybrid, that dashed line, is not as affected in comparison to the Eurasian watermilfoil line, the solid line. And so we can say on average, at all of these 2,4-D concentrations that were looked at, hybrid watermilfoil is responding less than Eurasian watermilfoil. And then on the right-hand side of the screen, we have a different study that was done by Hannah Hoff in the lab, who is expanding on Liz's work, but taking it down to that strain level. So she was also looking at the overall Eurasian versus hybrid response to 2,4-D treatment, but then also included 4 different strains of each taxa, 4 of Eurasian and 4 of hybrid. And so on this graph, we see our 4 Eurasian strains in black bars, and then 4 hybrid strains in gray bars. And then the dry weight is on our y-axis, and each of those lines in the panel is representing the grand mean of the given taxon. So the solid line is the grand mean for our Eurasian, and then the dashed line is the grand mean of that final dry biomass for hybrid strains. And then each panel represents control. Well, the left panel represents the control or the untreated plants, and then the right panel represents the treated plants. So as we can see, we still see that pattern of, on average, hybrids are responding less to 2,4-D, but even when we take a look at the control, we're seeing that these hybrid watermilfoil strains are growing better or more vigorously than the Eurasian watermilfoil strains. Those overall means are greater than the Eurasian. So this study that's shown on the screen was not done in Ryan's lab. It was actually done by some folks in the Wisconsin DNR, and it was led by Michelle Nault. And it's a really great field evaluation of this concept or finding that hybrids respond less on average than Eurasians to 2,4-D treatment. So what they did here was they took a collection of lakes that were containing Eurasian watermilfoil, which is shown on the top panel, and then they also took a collection of lakes with hybrid watermilfoil, shown on the bottom panel. And what they saw here, they also calculated the percent milfoil control based on the concentration after the fact and what they saw. And so we saw, or I guess they saw that the Eurasian watermilfoil got pretty good control with 2,4-D, between like 80 and 100% milfoil control in those lakes that had Eurasian watermilfoil. But on the flip side, we saw that the hybrid watermilfoil lakes didn't get as good of control when treated with 2,4-D. The lakes are kind of more in that 50% control range. So, a great field example of still seeing that trend that hybrids on average are responding less to 2,4-D treatments. This table is pulled from the EPA, and it's showing the fluoroperoxyphene benzyl, or maybe you know it as Percellacor, recommendations for Eurasian and hybrid watermilfoil. And as we can see here, those recommendations are generally higher for hybrid watermilfoil than Eurasian watermilfoil. And presumably this is because we have this understanding of hybrid watermilfoils are more vigorous growers and, on average respond less to 2,4-D or oxinic herbicide treatments. This is showing a genetic test that we can do. That's pretty simple. All of it can be done in-house in the lab with basic lab tools and techniques where we can distinguish the taxon and species from each other. So we can distinguish hybrids, Eurasians, and northern water milfoils from each other. And this is, we can easily distinguish those through an assay using the ITS region, the inner transcribed spacer region in the genome or the plant, and then running some gel electrophoresis on it using some enzymes, cuts them, and then we can see those banding patterns that are really distinct between the hybrid and the northern and the Eurasian water milfoil. So it's really important. We recognize that it's important to distinguish between taxon of Eurasian and hybrid water milfoil, because on average, our hybrid water milfoil grow more vigorously and are less sensitive to 2,4-D treatments, especially at lower concentrations. And this may also apply to other herbicides. However, we don't really have any great studies to back this yet, so it's kind of a likely situation. And this, we also saw varies by strain. So it's not true that all hybrids are resistant to all herbicides, and it is also not true that all hybrids are more resistant than all Eurasian. So if we go back to Hannah Hoff's study, and we look at the individual strain responses to 2,4-D, and not just the overall taxonomic responses, we do see that some of the Eurasian water milfoil strains in both control and treated conditions are growing similarly to some of the hybrid water milfoil strains. And then we also see similar patterns in the Wisconsin DNR study, where in our hybrid water milfoil panel, we see that some of those lakes with hybrid water milfoil responded just as well to the 2,4-D and got, around 80%, 90% of the milfoil control in that lake. So it's important to take into account the taxon differences that we see, but it's also really important to dive in a little bit deeper and look at the specific strain response, because there are differences there. We also see this in fluridone response, that there is variation by strain. Here highlighted are two highly resistant water milfoil strains to fluridone. The purple box is a hybrid water milfoil strain, and then my mistake. The purple box is the Eurasian water milfoil strain, and the red box is the hybrid water milfoil strain. So what this shows is that Eurasian water milfoil isn't necessarily immune to resistance. It can still exhibit resistance to a given herbicide. And there's also plenty of susceptible hybrid water milfoil strains to fluridone, as we can see in the red highlighted boxes. So this is kind of where we transition into more of the strain level of the talk and focusing a little bit in on my work that I've done in the lab. So as part of my master's work, I created, maybe a little bit biased, but I created a pretty cool app called MilfoilMapper. Maybe some of you have heard of it. I can include the link in the chat when we're done with the presentation. I can't quite do that in the chat right now. But yeah, I would love to share this resource with you. MilfoilMapper was created by me and with the help of Alex Badges over at Mazurk. Maybe some of you know him. Shout out to Alex. Thank you for all of your help. But MilfoilMapper is really a resource that shows all of the strain occurrences of water milfoil that we have across the US. So this is just a screenshot of MilfoilMapper, but it is an interactive database where users can click around, move around, modify filters, and interact with the database. I'm just going to highlight a few of the things that you can do with MilfoilMapper. So users can query by geography, and if they know a specific strain, they could type that into the box and pull up those results. Here I typed in this H. misgip 249 strain, and those are all of the occurrences shown on the map. And if clicked on for each lake, MilfoilMapper provides a list of strains and any available herbicide response information associated with those strains. So we have 2 strains in this lake. One of them has unknown herbicide response, and then this H. misgip 249 does have some known herbicide response information associated with it. And then it also shows the distribution of any strain and can print a list of lakes that that strain is present in using that list of lakes button over in the filters box. So this is a flowchart kind of working through how we envision users being able to use or MilfoilMapper. And so I'm going to walk through a few examples of this, but the main thing that we think is most valuable about MilfoilMapper is whether or not there's any herbicide response associated with a strain. And so you see this example on the screen. Maybe it's a little bit small, but hopefully you can zoom in or see it well enough. But this lake highlighted contains a strain called E. misgip 734, which is actually a really widespread Eurasian water milfoil strain. We find it coast to coast. And it is marked as sensitive to both 2,4-D and fluridone, which is great news because, there's really no concern about treating with this strain present in your lake, because really things should, work as intended and planned. However, we do have strains that are marked as resistant in the database to a given herbicide. Like this example that I mentioned earlier, this H. misgip 249 is resistant to fluridone. So we would advise against using fluridone or, the herbicide that is marked as resistant for that given strain in your management. So unfortunately, most of the cases for these strains is that we don't have any known herbicide response for it, but there's some other steps that we can take to kind of gain as much information as possible about those strains, even though we don't have known herbicide response information. So one thing that we would suggest is people can talk to other lakes that have the same strains present. So talking around with people, seeing what worked, what didn't work, how well it worked, etc. Maybe considering quantitative genetic monitoring. So seeing if there's any changes in strain frequencies over time, kind of giving an insight to, things that we might want to take into the lab and do some laboratory assays on. And then users or people can pool resources for laboratory or field comparisons, whether those resources be money or just kind of pool together a bunch of samples or efforts just to get a better understanding of the strains in their lake. So I want to point out some important nuances in these strain characterizations and those resistance and sensitive labels that we have on the strains. And I really want to point out that resistance is situational. So even though there is like a definite definition for herbicide resistance, these herbicide response curves vary, and it's not just a binary resistant versus sensitive. So this is because use patterns like rates and exposure time vary from place to place. And this could be due to regulations, so state to state. Regulations can vary of what concentrations are allowed to be used in lakes. This can also vary just simply due to hydrological functions of the lake or things that are out of people's control. And what I want to point out here are these dose response curves on the screen. I'll focus in on the one on the right-hand side of the screen, where we're looking at fluridone concentration on the x-axis and our final dry biomass on the y-axis. And then these are dose response curves for fluridone for a handful of watermilfoil strains, a mixture of Eurasian and hybrid. And we do see that there's a lot of variation in response. Not all strains are responding the same at each concentration, which is important to recognize and realize when we're making our management plans. And so, for example, if we take a look at that kind of middle cluster with all of those colorful strains in the middle of the distribution, maybe we would expect pretty good control at 6 parts per billion of fluridone. But then once we move back to 3 parts per billion, for whatever reason, if we were treating a lake at 3 parts per billion, then we might not get as good of control as expected and maybe even consider that resistance in certain cases. However, if we're to look at the strain, that red one at the bottom of the distribution, we'd expect to get pretty good control across the board, and there's no worry there. So they're just really pointing out that response variation across all of the strains and taking into account that, not all herbicides are going to be used at the same way across places, across states. So we got to keep that in mind. So generally, we see that there's more response variation at lower rates, lower herbicide rates than higher herbicide rates. And we can expect more variation in results when herbicides are used at lower rates. And this is common in aquatic plant management, especially because we are selectively treating for our target species while also keeping our non-target species at bay. So on the left-hand side of the screen is an illustration of the exposure time and concentration done in a study in the 90s by Grain and Westerdahl. And really it's just showing that as we increase our exposure time and our concentration, what that looks like with injury and control of these plants or a population of plants. And on the right-hand side of the screen is a dose response curve for 5 different watermilfoil strains, again, a mixture of Eurasian and hybrid, in response to 2,4-D treatment. So what we can see here is that these higher treatment concentration rates, for example, I think this is 2,000 parts per billion, we see a lot less response variation across these strains. And this is because, we're treating at really high concentrations and it doesn't take as long of an exposure time to get the control or death that we want to see. However, when you're looking at these lower concentrations, you need a lot higher exposure time to actually get that control that you're looking for because there is a lot of response variation. We can see that all of those lines end up differently and the strains are responding differently to this treatment of 2,4-D. So when we're planning for, looking at the general genetic herbicide response variation in watermilfoil strains, it's really important that we sample more genetic diversity to better understand the range of natural variation. For example, we don't just want to go off the boat dock and sample a bunch of plants there and say, yep, that's what our response to our herbicide treatment is, because that's not going to be a very accurate representation of the whole lake, first of all, and it's also not going to be a very accurate representation of all of the watermilfoil in the US. So we just want to make sure that we're sampling broad ranges, more genetic diversity to capture all of those responses that we may see. And with that, I'm going to turn it over to Greg.
Thank you, Ashley. So I'm going to pick up here at the genes level. So as Ashley showed, where we're at right now, we can track these strains and we can bring these strains into the lab and we can assign them an herbicide response value. And then as she showed with MilfoilMapper, you can actually track those strains across the landscape and predict whether or not depending on what strain you have in your lake, how your lake population may respond to a given herbicide treatment. And so we're pretty good at doing that at this point, right? We can bring these strains in, we can identify them as resistant or susceptible, and then we can bring in samples from folks. I'm not sure if that was mentioned, but folks can send in samples and we can tell you what strain you have in the lab, and then you can go to MilfoilMapper and compare where that strain also appears. And so that's great, but the one caveat there is that these populations of water milfoil are still able to sexually reproduce. And so when that happens, they're exchanging genes and the genes are getting shuffled up. And so individuals may appear as, well, they would be new unique strains made through sexual recombination, but they may inherit these genes that cause an individual to be resistant. And so we could go out and try to characterize every strain that's out there. It would certainly take a lot of effort. It would be really hard to do. And we would always sort of constantly be behind the ball because new strains are being made all the time through sexual recombination. But now that we know that we have some that are resistant and some that are sensitive, we can start to compare the genomes of those individuals and try to get down to the gene itself that's causing this resistance. And so in the future, what we sort of envision is that MilfoilMapper, rather than tracking clones or strains across the landscape, that we would be tracking causal gene variants that lead to some herbicide resistance. So that's sort of the premise for what we're trying to do in this sort of long term, right? And so a really good example of that comes from hydrilla management. And so as far as I know, this is the only assay that we have in aquatic plants, but we're working to bring Eurasian water milfoil to that point and some other species in the future, hopefully. But so I wanted to highlight this example here of hydrilla. This was done by another lab, not our lab. And what they found was, you can see in this dose response assay here, you can kind of think of the y-axis as biomass, like Ashley was showing earlier. And then you have your different concentrations of fluridone along the y-axis. And you see there's varying response between these 4 different strains of hydrilla. And so in this species, what they've actually done is narrowed it in to the gene that's actually causing that. So fluridone is a systemic herbicide, as was mentioned, and it's an inhibitor of phytoene desaturase, which is an enzyme that's required for plant proper functioning. And so the authors of this paper here went and looked through that phytoene desaturase gene, and were actually able to find single base pair mutations in this codon position that codes for number 304, the amino acid number 304. And depending on what single base pair mutation individuals have determines how individuals respond to these fluridone treatments, I should say. And so that's what I mean about getting down to the gene level. So ultimately, what would be great. So in this instance, actually, what researchers can do is, before you're treating a lake, you could go out and sample some hydrilla from whatever population you want to treat with fluridone. You can easily sequence this gene and look for these mutations, and from that, you can then infer how that population may respond to fluridone treatment. And so, like I said, this is really what we're driving towards in water milfoil, but there's a few steps that need to take place for us to get there. Okay, so where we started on this was we needed to know a strain. We needed to have strains that were resistant and susceptible to fluridone. And so, as Ashley showed earlier, we have this initial study looking at fluridone response among a bunch of different strains of Eurasian and hybrid water milfoil. And so to start looking for spots in the genome or that are responsible for this fluridone resistance, we took a resistant strain number 377 and a sensitive strain 1282 and crossed them together. And we can do that pretty well in the lab. We have cultures of these plants, and they put up these nice flowers that have both male and female parts. And so you can actually cross both ways between these two different strains, and we can make seeds from that. And then what we're really looking for is in those progeny, so all those seeds, we grew up and we kept them all separate, and we screened all of those progeny for 2,4-D resistance. And so what we're looking for is within all these brothers and sisters siblings, which ones are resistant and which ones are still susceptible to fluridone. So this was a study where we did just that. We grew up all those progeny, and you can see the sensitive baseline lake. Sorry, I didn't mention that. The sensitive one was from Baseline Lake in Michigan, and the resistant one is from Lansing Lake in Michigan. And so we have the baseline parent here. You can see in this picture. Hopefully you can see my mouse cursor. And then on the far right-hand side, we have the resistant parent from Lansing. And in between those, we have these progeny that either grow more similarly to the baseline, the sensitive parent, or the resistant parent. So, and then coming down to the left-hand corner of the screen, you can see this box plot. So the two red boxes are the Lansing parent, and this is just to show the spread of data for all of these individuals. So we have the resistant parent in both control and treatment, and that's to show that there really is no difference in this parent when you treat them with herbicide. And then in the blue box, you have the progeny, and in the green box, you have the susceptible parent, both in fluridone treatment. And so we can see that there's a spread among these progeny where some of them grow similar to the resistant parent, and some of them are more sensitive to the herbicide and grow more have similar biomass to the sensitive parent. And so in order to try to narrow in on genomic regions, what we can do is statistically compare between those two different groups. We call them bulks, and try to find areas where they're unusually different between one another. And so that's essentially how this works. We had a lot of help from these folks at Hudson Alpha. Alex Harkis and Josh Clevenger have been instrumental in helping us get these sort of studies going in this non-model aquatic invasive plant. And so we're extremely grateful for their help and assistance on this. Okay, so these are some results from doing those statistical comparisons. And I know it's a lot on the screen right now. There are 21 different plots, and that's because Eurasian water milfoil has 21 different chromosomes. So we've actually sequenced the Eurasian water milfoil genome, and we're able to assign reads or genetic variants back to the actual chromosome that they come from. But essentially, what we're looking at is all these little blue points is a data point. It's a spot where the two genomes it's a spot where the resistant genome and the sensitive genome differ from one another. And what we're looking for is areas in those resistant and sensitive progeny where they're unusually different between each other. So on average, these things are full siblings with one another. So on average, they should be fairly closely related to one another. And when they're not, it's an effect of the way that we separated them and compared them. So I'm going to zoom in here on the one chromosome where we're seeing a result that cues us in on somewhere in the genome that we think is associated with resistance. Okay, so this is chromosome 7 in the Eurasian water milfoil genome. And so on the top panel, all these green dots are spots that are different between the resistant and sensitive parent. We call them SNPs, and they're just single nucleotide polymorphisms. And this top panel with the green dots is showing the frequency of the resistant allele across the resistant individuals. And it's across the resistant individuals. 1 means that they have the resistant allele. Everyone has the resistant allele. 0 means that everyone has the sensitive allele. And so you can see here around this base pair, 30 million. So this is a whole chromosome. There's about 40 million base pairs in this chromosome. And around 30 million here, we have this peak where a lot of the resistant individuals are going up towards 1. If we drop down to the orange panel, it's essentially the opposite of that. So now we're looking within the sensitive bulk, among sensitive individuals. They tend to dip down towards 0, meaning they more have the sensitive reference allele. And then this final plot down here is what's really sort of the money figure. This is the difference between those. So you can see this is essentially just the resistant minus the sensitives in these SNP indices. And so the green and orange lines are our confidence intervals. Each point on here is a SNP marker, and the red line is the rolling average. And so essentially what you can see here is this peak around 30 million, where we have a large difference between resistant and sensitive bulks, meaning that there is likely a gene in that region that is associated with or causing herbicide resistance. Interestingly, I'm kind of presenting these a little bit backwards, but interestingly, this is not where the PDS gene is. So if you remember back to the hydrilla example, we talked about mutations in the actual target enzyme gene. That is actually on chromosomes 10, 11, and 12. And we also looked for mutations in that. I won't go through this table in detail just for time, but essentially what we found was that there are no mutations within that gene that are associated with herbicide resistance. Additionally, another way that individuals could be fluridone resistant would be in overexpressing that PDS gene. And so we also looked at gene expression between the resistant and sensitive individuals in both control and fluridone treated environments for how much PDS gene was being expressed. And we also saw no differences there. And so we've narrowed down the entire genome to this one chunk that's around 3 million base pairs. I know that sounds like a lot, but the Eurasian watermilfoil genome is like 800 million base pairs. So it's quite a bit narrowed in from where we originally started. And within that area, we have a few genes that we can start to investigate and look into. Just real quickly, I know I'm sort of getting close on time here. We constructed a second mapping population using that one strain in Michigan that's fluridone resistant that Ashley showed on MilfoilMapper. Tends to be fairly widespread within Michigan. We're constantly getting more lakes in that have that strain in it. And that is also a fluridone resistant mapping population and that we have created. And interestingly, we're seeing a very similar peak. So on the left here, we have that first mapping population, and on the right, we have this new mapping population. And so this suggests that maybe these two strains are fluridone resistant in similar ways. And that would be really great for us because we ultimately want to develop markers for this, where we can go out and screen these populations. And this means if individuals tend to do it the same way, we only need one marker that works. But of course, new ways could always arise, and we would have to develop new markers. But so like I said, that's what's currently in progress for our long-term sort of plans. Ideally, we would be able to narrow in on this chunk and look and find the actual causal gene. So like I mentioned in hydrilla, like this is the gene we know, like this exact mutation causes fluridone resistant. It would be great to get to that and water milfoil. But if we can't quite get to that level, one other thing that's in progress is developing some markers that are in this region that seem to be closely linked with that causal gene. And we may be able to use not necessarily the causal gene, but markers that surround it to characterize, test uncharacterized strains and assign herbicide resistance. And we'll begin to test those markers shortly here in other populations and hopefully see what we see. Okay, so just a recap, we talked about the short term, building this catalog of strain information. So Ashley shared with you the MilfoilMapper, and in the short term, we can use that to determine whether or not we know anything about a strain and try to predict how a late treatment may go if we know things about a strain. In the long term, we're trying to get down to this genetic basis of resistance so that we can ultimately track genes instead of the clones or strains themselves. And hopefully our goal is that this acts as sort of a model for these types of tools in weed management. So like these types of tests that I've been talking about are fairly common in terrestrial agricultural populations, but they're not very common in aquatic plants. Well, they're not common. They're not common in aquatic plant management, and they're rarely employed in other types of weed management, maybe in natural lands or rangelands and things like that. And so hopefully this shows that it can be done in other systems other than just agricultural weed management. So we have a lot of people to thank. Mostly our funding comes from the Army Corps of Engineers, but there are several other like state agencies, lots of people who have sent samples. Ray Newman was instrumental in getting a lot of these things. Like I said, Alex and Josh have really helped out with the bioinformatics type stuff. And I know we're tight on time, so I will end it there. Thank you.
I'll stop sharing the screen too. Perfect.
There we go. All right. Thanks so much to both of you for all of the wonderful information. I'm going to have the slide up for maybe about 5 minutes, and then I'll minimize it so we don't have to just stare at a QR code. But you can enter your questions for Greg and Ashley at slido.com. Use the event code MilFoil, or if you have a phone or mobile device, you can scan that code. If it's not working for you or you'd rather use chat, go ahead and put it in Zoom chat and Michelle will transfer it over. The bonus of using Slido is you can upvote the questions. So I'm going to go ahead and dive right in and we'll turn off my screen share here in a little bit. But the first question for you is wondering what the cost and availability of the genomic testing tends to be. And I might tie that in with a question right below it, which is also wondering, like, where are the places that you can get that kind of testing done? And is there a list someplace? Is there a turnaround time? Like, how does that work?
So yeah, we do a lot of like the strain identification tests. We do a lot of that in-house, and I would be the person to contact if anybody is interested to send in samples to talk in further detail about how to do that in your lake and whatnot. The general cost, we usually say to collect around 20 samples from a lake, and that is around $1,000. So maybe cost prohibitive in some cases, but it is a fee for service, and we have some dedicated funding, but that's pretty limited and state specific. But yeah, a lot of that happens in-house, and contact me for more information if you want that done.
And as far as I know, we're the only ones that are doing that. So there are no other facilities. You would have to contact Ashley to have your strain characterized.
Yep.
All right. Great. Thank you. Okay. The next question is wondering if there's any differences between water milfoil strains based on geographic location. So the example they had was Montana versus Minnesota, but maybe on the MilfoilMapper, there's some other indicators for based on what's on that map.
Sure. So I guess there are different strains present in different states. We do have some Eurasian watermilfoil strains that, like I mentioned, are very widespread and found coast to coast. So those are kind of shared across states. And in terms of herbicide response, those strains, can vary state to state based on herbicide treatment history in that given state. So, for example, Michigan has some pretty heavy fluridone use and has been using fluridone for a long time. And that's where our fluridone resistant strain, that Town Line Lake strain, or H. Misgip 249, whichever you remember it as from the talk, that's where that one's found. It's pretty widespread throughout Michigan. It gets found in 10 plus lakes. So far, we haven't found it outside of Michigan. And those hybrid strains, for whatever reason, tend to be pretty restricted within the state. Yeah, so hopefully that answered it. Greg, if you want to add anything, go for it.
Oh.
All right. Could either of you talk about impacts that you know about using some of these herbicides that are shown to work with hybrid water milfoil on other aquatic life?
I can speak a little bit about that. We haven't done any of those types of studies in our lab. But what I will say is when you increase herbicide concentrations in a lake, you're going to get more non-specific effects, right? So there are lots of different plant species that are living in these aquatic environments. And the reason why most of these aquatic herbicides are used at the concentration they're used at is to try to specifically affect Eurasian water milfoil without killing those other plants. And so if you increase your concentration to maybe more effectively control the hybrid water milfoil, you're probably more likely to damage those native aquatic plant populations. Yeah.
Right. Okay. Next question. So we've seen a lot about eDNA lately, and this next question follows along those lines. So there's a lot of talk about using eDNA to identify early detections, and if a species is present, is that tool a possibility to also identify down to a strain?
Yeah, that's a good question. So a long time ago in the lab, we did do some stuff with eDNA work. And back then we were just trying to distinguish between hybrid and Eurasian, just detecting whether or not you had hybrid individuals in a lake water sample or not. We haven't done much in that realm lately. I assume that you could come up with a test to determine these things. But what I will say is typically these types of eDNA assays rely on like one to a handful of like really conserved genes. And what those will tell you is the species or the taxa level. And they can't tell you necessarily the genotype level. And that's because DNA tends to degrade in these environments fairly rapidly. And so in order for us to genotype, we need to assay several spots in the genome that are variable within a species and not necessarily the more conserved genes that are present across species in order to, so a long answer to not currently. I don't know if there is a way to do that. I haven't seen anything out there that does that.
Great. All right. All right, I'm going to ask this next question, then I'll shut off screen share, which might change your screen a little bit, so don't be alarmed as that goes. The next question is wondering if there's an assumption being made that the gene affecting sensitivity would be an herbicide-resistant gene versus a gene that causes vigorous growth. And this person mentioned that this was an idea discussed in Hannah's paper.
Yeah, that's a good point. So the phenotyping that we're using in these assays is biomass, and so currently we found, although Ashley has been working on some other ways to phenotype for herbicide resistance, but currently the most reliable phenotype that we can measure for herbicide sensitivity is how well does it grow well exposed to herbicide. And we can control for that by having control and treated plants. And we can look at the difference in growth between the exact same clone of, because we can vegetatively propagate them, right? We can look at the difference between how they grow in the control and the treatment, and we can measure that. And so hopefully in comparing between control and treatment, we would control for differences in growth rate, but it's entirely possible that vigorous growth is smuggled in there, and that when we do these types of genetic mapping, that we may be detecting genes that cause growth differences between them also. Yeah.
All right. So if there were a lake where there was not a fluridone resistant strain present, this one person is wondering, they say it seems like an aggressive lake-wide treatment for Eurasian watermilfoil followed up by regular monitoring or spot treatment would slow the formation of a new resistant milfoil strain. Does that seem correct to you?
It's tricky. That would be true if you could ensure that you killed every plant in that lake and that no plants were going to be coming back into that lake. Otherwise, what we know from other weed management, so terrestrial weed managers and agricultural weed managers have been dealing with this for a long time. It doesn't tend to matter how high you hit them with, what type of doses you use. Sort of nature finds a way, and new mutations arise in a population. New strains come in from outside that can handle that type of herbicide dosing. And so I guess I would caution you from thinking that, like, well, if I just hit it harder with this herbicide, I won't have a weed problem, because that has not worked in other systems, and I don't think it's likely to work in the aquatic environment either.
All right, we probably have time for one or two more questions. Next one is wondering about Procellacor and if that's one of the herbicides that you've been able to look at in terms of resistance, or if it is it on your radar?
It is on the radar, yes. We actually are, well, I guess we have applied for a grant to hopefully look at some responses to, just getting the existing genetic variation or herbicide response variation to Procellacor treatments in the Great Lakes Basin. And then we also have a Michigan specific grant where we're going to be doing that. So we will be getting some Michigan specific results on that soon. And hopefully we can expand those results to the basin as well if we end up getting that funding. So it's something in the works, something definitely of interest for a lot of folks, for us too. So it'll be great to start being able to share those results in the future with you guys.
Great. Sounds like we can get a future webinar planned too then. All right. Thank you both. I see it just hit 2 o'clock. I'm going to share my screen real quick for some final housekeeping items. So I mentioned that we did record this webinar and we'll have it up on our YouTube channel. Again, it takes a week or so for us to get that transcribed and ready to go, but you can find our YouTube channel and all of our past webinars at z.umn.edu/AIStube. You can watch your inboxes for a notice when that recording is ready for you. You should see a link to a survey when you sign out, and we would greatly appreciate it if you took some time to answer those questions for us about the today's webinar. I also do have some contact information up on the screen. So for those of you who are now ready to email Ashley to ask more about how to get your strains tested, you can see her email up on the screen there. If you have more general questions about like the webinars or webinar series, then you can reach out to us at AIS Detectors at umn.edu. And with that, I will say my final thanks to you, Ashley, and to you, Greg, for joining us and sharing all this great information about managing hybrid water milfoil. There was a lot of interest and a lot of really great questions. We appreciate you being here and sharing all of your great work with us.
Yeah, of course. Thank you so much for having us. We really appreciate you setting up this webinar and having us present. And if any of these questions didn't get answered, please feel free to reach out to me. Like Megan said, my email is up on the screen. So definitely don't hesitate to ask those questions that didn't get answered.
Thank you.
Great. All right, great. Thanks again to both of you, and thanks for everyone for joining us. We hope to see you at our next webinar, which will be in May. And this one is going to be about rusty crayfish management. So you can check for links to register for that one next. Thanks to everyone. Have a great rest of your day.
Thanks, you too.
All right.